Okay, we'll be starting with the first test for coliform, that is member filter technique. Membrane filter technique. So first we'll write down the points, the most important points from here. So the water sample is poured on the membrane filter on the membrane filter. This filter membrane filter is very fine, or you can even write it very fine. The pore size of this filter is very small. So it is so small, it is so small that even water cannot go freely through it. So even water cannot go freely through that member or that filter. Now on this filter, the specially for food specially for coliforms is placed. Food is placed on this membrane, and it is once the food is placed on this member, the water is transferred from this filter. There are certain number of coliforms which will get attached to this member, same as we have seen for suspended particles. See, there is one filter paper. So in this we'll call this as membrane filter. Membrane filter. So water is poured on this filter, and on this filter paper there is some food specially for the coliforms. Okay, so this is the food. Now what will happen? The coliforms which are present in the water, they will start eating this, or we can say they will surround it here. Once the water is penetrated, or once the desired amount of water passes through this membrane filter is taken for incubation. Okay, this membrane filter is taken for incubation. See, if you remember, for determination of suspended particles, we have used a filter paper. That is, we have passed the water through the filter paper, and that filter paper was then transferred for oven drying. So this incubation is similar to oven drying only. See, what is the main difference between oven drying and incubation? See, this incubation, it has come from a device which is called as incubator, and this incubator can do both the functions. It can do heating also, and it can do cooling also. This heating is also done by oven drying. So you can see now the difference between this. Oven drying is only used for heating, whereas incubator can be used for heating also for cooling. So based on the requirement, we can use the incubator for heating or cooling. In this case, we will use it for heating. Okay, so this is the difference between incubator and oven. Now, so our membrane filter is taken for incubation now, and after this, with microscopic analysis, number of visible dots are found on the member. Okay, so with the help of microscope, what we'll do, we'll take this membrane filter, and we will see microscopically. If we are able to see some dots, then these dots will represent the presence of coliforms. If dots are not present, that means there is no coliforms present. If dots present, coliforms present. Okay, and this one dot, or you can say the one dot, this represents millions or billions of bacteria. Millions, or you can say billions also. Okay, so this number of dots are identified. If the number of dots are present, that means the coliforms are present. If coliforms are present, then pathogens are present. So this is the method which is used for the determination of coliforms. Okay, now in the next video we will see MPN test.